Hello, can somebody confirm that you can hear me?
Yes, we can hear you, Professor.
Yes.
Great. I need my glasses, but we'll begin. All right, you should be able to see my screen now. Come on. Now I can't read this. It's February 29th. Let's see if I can find my glasses. Here they are. And we should be talking about Chapter 6, Microbial Growth. Any questions? Tonight, there is no lab scheduled. You should be working on your unknown project. I will be logging in for 15 minutes just to answer questions, but unless there are a lot of questions, I'll only be there from 6:30 to 6:45. So if you have any questions, make sure you log in at that time to ask them, and I will answer them. When the last question is answered or at 6:45, I will log off. Any questions about what we're doing today in the lecture or in the lab? Sorry if you heard that. That was my stomach really growling. All right, no questions. So let's begin chapter 6. And remember, in the lab, you can show up if you have questions. If you don't have any questions, there's no reason to show up. And then at 6:45, I'll be logging off. All right, let's go to chapter 6. It seems to me we started this.
Yes, I believe my last notes were about
Boy, we weren't at page
E.
30.
coli.
We were not this far, I'm sure. So I didn't mark it. That's not good. So can you tell me where we got? I do, I don't remember discussing that. I don't remember discussing that either. Maybe we didn't start chapter 6.
We didn't.
Okay.
I have amphibolic pathways is the last note that I took.
So we didn't quite finish chapter 5.
My last notes were anaerobic respiration, but
Okay. No, I do
Correct.
remember talking about amphibolic pathways. Let me take a look and see if I've got a marker on chapter 5. Let's see if that goes page 70. So we did discuss this. Oh, there it is there. There's my marker. So there's only one more topic I need to talk about in this class, in this chapter 5 lesson, and that is photosynthesis. Photosynthesis is the conversion of light energy to chemical energy. In photosynthesis, chemical energy may be used to convert atmospheric CO2 to glucose, and this is called carbon fixation. All life, well, almost all life on the planet is dependent on photosynthesis because almost everything we eat comes about because of photosynthesis. And you might argue, well, I don't eat green plants. I eat a cow. I eat beef. And what do you think the cow eats? The cow eats grass. So the cow is only alive because of photosynthesis. it is true that there are some places in the world where you might have life without photosynthesis. And generally the only, what do you call that? Niche that is around without photosynthesis are the deep sea vents. They have an abundance of life, and it's because of the chemosynthetic organisms living around the volcanic vents. And they're producing glucose, which then the animals eat them, meaning the chemosynthetic, mostly bacteria. Any question about any of that? All right. Photosynthesis, of course, performed by green plants. You all know that. It's also performed by algae and some bacteria, such as cyanobacteria. And all of these are green, but they can be other, excuse me, I'm about to sneeze here. Sorry, but if I blow my nose, I might not sneeze. And don't know where that came from. All of them are green, but they don't have to be. There are photosynthetic pigments which are not green. Obviously, in green plants, it's chlorophyll, and that is a green photosynthetic pigment. Although if you ever see the leaves when they're yellow, that is a photosynthetic pigment also. When the leaves turn red, that is not a photosynthetic pigment. And we're not going to talk about that, but there's red algae, the photosynthetic pigment is red. There's brown algae, the photosynthetic pigment is brown. And in the cyanobacteria, it's actually a blue-green color, the photosynthetic pigment. Anyways, photosynthesis is a mechanism for synthesizing organic compounds, and green plants and other photosynthetic organisms can use photosynthesis to make ATP as well as make glucose. Photosynthesis only occurs in photosynthetic organisms. In photosynthesis, they always make ATP and NADPH. That's very similar to NADH. However, oftentimes in photosynthesis, the ATP and the NADPH that are made in photosynthesis are used to synthesize glucose from CO2 and water. And that might be why when they have talked to you about photosynthesis before, they'll say something like CO2 and water makes glucose, and they never talk about ATP because it's used as an intermediary to make glucose. But it is made in photosynthesis, in all forms of photosynthesis. There are actually two types of photosynthesis. There's the cyclic photosynthesis and the non-cyclic photosynthesis. When they've discussed photosynthesis with you before, I'm sure it was the non-cyclic photosynthesis that made oxygen. And so this would be oxygenic non-cyclic photosynthesis, where 6 carbon dioxide, 12 water molecules in the presence of light make 1 glucose, C6H12O6, 6 oxygen molecules, and 6 water molecules. Sometimes you can have the 6 water subtracted, and then you subtract 6 here. I prefer it shown this way because in photosynthesis, all of these oxygens come from water. 12 oxygen in water, 12 oxygen atoms in 6 molecules of oxygen. Somebody has their mic on. Let me see. Oh, can't get it. Just stop the share to take a look at that. All right, it's been turned off now. Good, good. So all of the oxygen, all of the oxygen come from water. None of the oxygen come from carbon dioxide. All of the carbon dioxide is converted into glucose, and some of it is converted into water. Any questions about any of that? Now, there is another non-cyclic photosynthesis, and it does not generate oxygen. And this is the summary equation for anaerobic photosynthesis, where we get carbon dioxide and sulfur, or actually hydrogen sulfide gas, in the presence of light to make glucose. And it doesn't produce oxygen, it actually produces sulfur. And you don't need to know this reaction, so you can just skip it. However, there's another photosynthetic reaction, and it does not make glucose. With light hitting the green plant, it makes ATP from ADP and inorganic phosphate. And that is cyclic photosynthesis. This one has probably never been taught to you, so I'm going to teach it to you now in just a few slides. All photosynthetic organisms actually can perform both photosynthetic reactions, meaning they can make glucose, either this one or that one, and then they can also make ATP. They will do whatever photosynthetic reaction they need. If they need glucose and it's a green plant, it'll engage in this photosynthetic reaction. And obviously, if it's a bacteria that doesn't make oxygen, it'll engage in this one if it needs glucose. If, on the other hand, the cell needs ATP, it'll engage in cyclic photosynthesis. Cyclic photosynthesis is when light excites two electrons from chlorophyll. And that actually happens in both non-cyclic and cyclic photosynthesis. In non-cyclic photosynthesis, the electrons flowing out of chlorophyll are replaced from the electrons from water. And in cyclic photosynthesis, the electrons flowing out of the chlorophyll move to a protein in a short transport protein, meaning in a short electron what do you call that? Transport. Drawing a blank here, what that's called. It's
Is it the electron transport chain?
Yeah, electron transport chain. Thank you. It's a short electron transport chain, and it's a bunch of proteins, once again, in a membrane. The electron flows out of chlorophyll. Let me blow this up. When light hits the electrons move out of the chlorophyll, moving into this protein in the electron transport chain, and then moving into this protein, and then moving from that protein into this protein, and then from this protein, the electrons return to the chlorophyll. So the electrons finish where they started. And that's why it's called cyclic photophosphorylation or cyclic photosynthesis, because the electron travels in a cycle or a circle. Okay. The point of the electron transporting from one protein to another is that when it moves through this electron transport chain, it gives this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. the cell then uses this a hydrogen ion gradient, and it's twofold, both more positive charges here than there, and then more hydrogen here than there, and that makes a potential energy difference. The cell uses this potential energy difference to generate ATP. The hydrogen ions can only flow through the membrane in one spot, and that's right here, this channel protein, which is a ATP synthase protein. And when the hydrogen ion flows through this protein, it gives the protein the energy it needs to add phosphate to ADP to make ATP. Now, this should be very familiar to you because we've seen it before, right? We saw it in aerobic respiration, and the same thing happens in anaerobic respiration. And that is the electrons are moving through a protein, causing the hydrogen ions to be pumped from one side of the membrane to another. And this electron transport chain is very similar to the electron transport chain we just recently saw in aerobic respiration. It is obviously different. For one thing, there's only 3 proteins in the chain, plus, I guess, this one, which is more than one protein. It's a whole photosystem. So there's a bunch of proteins in this photosystem one. But the electron moves from, well, I don't know, from this protein to that one, to that one, to that one, and then back to this one. That's cyclic photosynthesis. Any question about cyclic photosynthesis? Whoops, didn't mean to be there, meant to be here. In cyclic photosynthesis, the cell and all photosynthetic cells can do this. They can make ATP directly and then use the ATP for their energy needs. And the alternative is if they need glucose, they engage in non-cyclic photosynthesis. All right, so briefly, let's go over this summary reaction. For making ATP production, the energy source can vary. We just talked about the energy source can be light from the sun, and the light from the sun causes the electrons to move. And the electron carriers can vary. In photosynthesis, the electron carriers are NADP+, which is very similar to NAD+, which we saw in aerobic respiration. And the other one is FAD, which we also saw in aerobic respiration. And then the final electron acceptor can vary. For aerobic respiration, oxygen is the final electron acceptor. For anaerobic respiration, it's a molecule other than oxygen, such as nitrite, nitrate, shown here, sulfate, or iron. For fermentation, the electrons flow to an organic chemical compound, usually pyruvate, in fermentation. And then the fourth, which isn't shown here, the electrons flowing from cyclic photosynthesis actually end up at the chlorophyll, the place where they began. And that is the final electron acceptor for cyclic photosynthesis. I don't know if I mentioned that the electron donor can vary. For aerobic respiration, it's usually glucose, but it could be any carbohydrate or any other food molecule. And the electrons flow from that. I did mention with glucose, the electrons flow and they'll be carried by NAD plus and FAD to generate ATP. Any question about any of this? All right, if not, that's it for chapter 5. Let's begin chapter 6. Why did I stop the sharing of the screen? Sorry. Did I stop the lesson? Is anyone there?
I'm
But
here.
we're still here.
Still here. It's
Yeah.
being
That's really
recorded.
weird. I think what I did was I
course.
accidentally stopped the sharing the screen.
I believe you're still recording.
Yeah, I'm still recording. I didn't mean to stop sharing the screen, and I certainly didn't mean to stop recording, just to be certain. I have to stop sharing the screen to see that, but all right, let's continue with chapter 6. Why is that all dark? Oh, because I'm still showing the screen. All right, let's end that and probably save it. Chapter 6, microbial growth. Some major goals and rough outline are discussed in this slide. Of course, always know the terms for the chapter. What's a psychrophile, a psychotroph, a mesophile, a thermophile? What's a hypothermophile? What are acetophiles, halophiles? What's an aerotolerant anaerobe, facultative anaerobe? What is binary fission? What are reproductive spores? And then what are endospores? Understand the various requirements for growth, and we're talking about microbial growth. Understand the 4 phases of growth, and then lastly, be able to compare the different methods of measuring microbial growth. Any questions about what we'll be discussing? All right. When we talk about microbial growth, it refers to an increase in number of the cells, not necessarily an increase in the cell size. I used to teach middle school students.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
I used to teach middle school students, and when we talked about growth, obviously we weren't talking about an increase in their numbers. But with microbial growth, that's what we mean. All living things, including microbes, have various requirements for growth. There are physical requirements, and these include it has to be within an acceptable temperature, an acceptable pH, an acceptable osmotic pressure, an acceptable salinity, and that's related to osmosis. And there has to be chemical requirements for microbial growth. There has to be a source for the microbe to get carbon, a source of nitrogen, a source of sulfur, and a source of phosphate. Now, why is there a source of sulfur? Obviously, it would be a trace element, would be my guess, because you don't need very much sulfur. You do need some. There's trace elements, and these would be something that you don't really necessarily need to supply to the cell, because you usually get trace elements directly from the media or the water that is used in the media. And then there's various oxygen requirements and organic growth factors, which may be required by certain things. An example would be Streptococcus pyogenes, which causes strep throat. You have to supply certain organic growth factors. And what we do in microbiology is we just put in red blood cells, and then the Streptococcus pyogenes can grow on the petri dish when it's supplemented with red blood cells. Any question about any of that? All right, each species of organism has a minimum, a maximum, and an optimum growth temperature. We can classify microorganisms into the groups on the basis of their preferred temperature range. And we'll talk about each of these, but the first is a psychrophile, psychotrophs. Both of these are cold loving. Psychotrophs a little warmer. A mesophiles, this is body temperature, like around 37 degrees Celsius or 98.6 degrees Fahrenheit. Thermophiles, what we would consider hot. And then hypothermophiles, very hot. And this is very close to boiling water. So for the physical requirements, temperature is one of the main physical requirements. All organisms grow within a range. For the psychrophiles, they grow best around 10 degrees, 11 degrees Celsius. They have a range, something like a minus 10 degrees to 20 degrees. And they can produce antifreeze to help them grow if they're very at a very warm temperature. Excuse me, that's not very warm temperature, very cold temperature. They can produce antifreeze to keep them from freezing. For very warm temperature, they can produce something else. What is that called? You put it in the car and then the car runs better. Is that in the winter you add it, or is that in the
Antifreeze?
summer?
Antifreeze?
Yeah, antifreeze can elevate the temperature, but I'm talking about what do you put in the car so it can tolerate a higher temperature?
Coolant?
A coolant. What's the coolant? Do you remember? No, okay. Yeah, you can add a coolant, and the bacteria can produce a coolant, and that's why it's surviving at 20 degrees, but it prefers really around 10 degrees Celsius. A psychotroph is an organism that does better, grows better. It's slightly elevated from the psychrophile. And this would be something, well, actually the psychotrophs, you can find them growing in your refrigerator. And that's why your food will rot in the refrigerator. It may be a week longer than normal, but still psychotrophs can grow in it and then either degrade the food or produce toxins. So you have to be careful about that. And psychotrophs are ones that do best around 20 degrees Celsius. It's a little warmer than psychrophiles. Psychotrophs are a little warmer. Psychotrophs also are the organism that's capable of reproducing. We're talking about microbes, so I tried not to say the word female, but you could call it like a female bacteria. Mesophiles are organisms that grow best around 37 degrees Celsius, which is the temperature our body is at. And they grow best at that temperature. They have been only discovered fairly recently. So those mesophiles aren't discussed that well. But if you get a pathogen and it's growing inside of you, it's likely to be a mesophile. And that's because it can grow very well at our temperature. However, you could perhaps see 1 in 100, 1 in 1,000, something like that, colonies develop from a psychotroph that has 4 different cells. One of those 4 being one that can grow at higher temperatures. And obviously there's some that would grow at lower temperatures. The majority grow at your average. Thermophiles are organisms that grow best between 60 and 65 degrees Celsius. if you think about the rotisserie at Safeway or some other grocery store, and it's just going around and around, once it starts drying out, you might want to pass on it because there are psychothermophiles which can grow at this temperature. And if they grow at this temperature, they'll grow quickly because this is the right temperature for them to grow at. And you don't want people like Gordon to spread that virus to other people. Any questions about any of that? All right. As I stated, the thermophile was known when I was an undergraduate in microbiology. The hypothermophiles was not known when I first took microbiology. I'm not sure how long it's been around, but hypothermophiles came out shortly after the discovery that archaea have two membranes. And I should try to show you some pictures of that, but I just don't have it. So try and use your imagination. If you want, I can say, yeah, that's good, or no, keep practicing. Any question about any of that? All right. So hypothermophiles grow best between about 92 degrees Celsius and 100 degrees Celsius. We could, under certain circumstances, be whole body irradiated, killing off the white blood cells and the lymphocytes, white blood cells, or whatever you want to call it, the lymphocyte blood cells. You should come to realize that I'm showing a range here, and this is just an approximation for what the range is. There could be one which takes much longer than what we have. There could be one which divides at a higher temperature too, and it's just not very common, in which case it would be a plus for the temperature requirement hypothermophile. But the range is something that is not rigidly defined. For mesophiles, for example, all the mesophiles that live on the skin, unless you're really close to the nose or the mouth, they're not going to grow at this temperature. They're going to grow best at an elevated temperature. Okay? Any questions about any of that? All right, if no questions, let's move on. I briefly want to talk about food spoilage. Most food spoilage occurs when the food is left at too high a temperature for too long. Let me see if we can blow this up because it's kind of small. The danger zone, which you don't want to have food there for very long, is between about 40 degrees Celsius and 130 degrees Fahrenheit. Now, this is combining the different temperatures, and I'm only going to talk about it, but you could look at it with nanometers or micrometers, and that would be used with the proteobacter, meaning they're on a runs right now. So if food is left at the red or the lighter colored red or something like that, then food can spoil, and food can spoil too quickly. And then when the user eats the food, he won't notice that it has bacterial growth down the bottom of it, and it has bacterial growth in the chocolate Reese's snowman. Any question about any of that? Come on. I'm not seeing my mouse. That might be it, but I'm not able to advance. Right click isn't working either. How bacteria could be placed in an environment suitable for growth underneath the light is that both tubes containing the correct reagents could be put into one UV machine and then irradiated, and then you put in the second cells and irradiate those. But it could be done that way. That's a little lengthy way of doing it. The principal reason why food spoilage rate occurs is because the food is packaged in too large of a volume, and so it never cools before the bacteria double. Another thing would be if you package the rice up and it's a smaller volume, but the rice itself takes a long time to become soft. And this is showing you a chart of that. All right, another physical requirement of all living things is pH. Most bacteria grow between a pH of, let's say, 6.4 and 7.4 degrees. Any questions? If there are questions, you should ask them so I know to repeat or slow down or something. Because most bacteria grow in a specific pH, we can use a lab culture of the appropriate organisms. Most bacteria grow around pH 7. They can go down to pH 6 and as high as pH, let's see, 7, no, pH 8. And they can have a larger range than that, but that's for probably most bacteria. Their pH range is between 5 and we'll just say 15. Molds and yeast are a little separate. They grow best in a little more acidic environment than a bacteria, and they grow best between pH 5 and pH 6. Acidophiles, on the other hand, grow in very salty environments or very acidic environments, And if you take them out of these in a salty environment, they may not grow. Acidic environments have acidophiles growing in them, and one acidic bacteria growing in a volcanic vent is when it's in a volcano protein.
So I have a question then. Volcanic environments, what exactly in the volcanic environment would be an acedophile?
It could be like a deep sea vent. If it's putting out an acid, it will be an acidic environment. Another one would be when we're digesting nitrogen. Some of them could be an acidic protein, meaning requiring a lower pH. I can't think of another example other than the one I gave, and that is a volcanic environment. It's acidic, and so acidophiles grow there. And the acidic environment of a volcano is sometime between pH 0 and pH 3 when we're talking about the organisms that grow in your stomach. What is that? Enterobacter. What is that name? Xylophile? No. It'll come to me. Anyways, there's an acidic protein that can live in acidic environments. And I'm not sure how acidic we can go with the cell still being alive, but at least it goes down to at least pH 1. And that would be as acidic as your stomach. And if you
Okay,
want to
thank
know
you.
something like 1 hydrogen ion and something like 10 to the 6 water molecules. And if you remember, pH is a measure of the hydrogen ion concentration. Another physical requirement of all living things is osmotic pressure. Microbes obviously require water. All living things cannot survive without water. And we are, people, are mostly water, about 70%. Any question about that part? All right. Microbes require a specific osmotic pressure environment. High osmotic pressures can remove the necessary water from a cell. And let me see if I can blow this up. If we were to put the cell in a hypertonic solution, yes. Not sure why that's asking that. Oh, come on. For some reason, I can't blow this one up, so I'm just going to go like that. I'm going to guess that I've got a mistake on this slide, and it's not, it's interfering. Gosh, that's not blowing up. Oh, here we go. I couldn't get it to blow up. If you put a cell in a hypertonic solution, there's more solute outside the cell than water inside the cell. And the water will move from inside the cell to outside the cell. This is only in a hypertonic environment. The cell wall can help with this, meaning it does offer some protection, but it's probably not enough when you're in an environment that requires a high osmotic pressure, and then you get another couch cushion. It's pretty hard putting in the second couch cushion. A hypertonic environment is when the water will move out of the cell, and that will shrink the cytoplasm. Here we see the membrane of the cytoplasm is definitely shrunk away from the, come on. It's definitely shrunk away from the agent that's causing problem for glucose metabolism. Any question about any of that? Okay, because some organisms can't grow in high salt or high sugar, we can use osmotic pressure to preserve food. And we still do that today, such as sealing a vacuum jar of applesauce. That is using osmotic pressure to prevent the death of the cell. This may not kill the cell, although it's not going to be good for the cell. It can't perform all the metabolic reactions it needs to because cytoplasm has decreased so much. The point was high osmosis can be used to preserve food, such as salted meats, molasses, salted fish, and then the one we've talked about previously, not egg rolls, what are those called? Geez. A little candy, saltwater taffy, okay? If you were to put a cell like this and it was a human cell, like a red blood cell in water, this water would move into the cell. No, water would move out of the cell. And water would continually move out of the cell until the washing machine and dryer stop, or until the timer runs to 0. Okay. This slide is showing you isotonic solution where there's no net movement of water into or out of the cell. So water is not moving, usually in an isotonic solution. You could talk about movement by sliding over the agar, in which case that is not true motion. Okay, any questions about any of this? Let me shrink this. Halophiles are organisms that like living in high salt. There's 2 types, an obligate halophile, and this requires salt concentrations to grow and for the enzymes to bond correctly to their substrate. There are also facultative halophiles, and they do not require high salt to grow, and they can even tolerate oxygen less than 72%, or excuse me, less than 15%. Some facultative halophiles can grow when the oxygen is less than 2%. And of course, that would be lethal to us. A hypotonic solution is one with low osmotic pressure, where there's more water outside the cell than is inside the cell, and that will create a difference along the cell membrane as to what it needs. And every time somebody with a banner crosses the lab here, if it splits into 2 files, you know that that person did not perform lab 3 correctly. Any question about any of that? All right. I noticed some people looking at the chart, and what you just do is you find out what your osmotic pressure is, what are the possible bacteria, and then try and figure out which bacteria may have caused the disease. Usually you have to run some biochemical test, because although this gram stain is very useful and helpful, let's move on. It's not perfect because it allows other molecules to leave or enter the cell membrane. Oops, I'm going the wrong way. All right, any questions about any of that? With high salinity, we get more than the shrinking of the lab coat of the 6-year-old student. Let's go back a bit. So if the osmotic pressure isn't correct, the protein will unfold, losing its activity. If it unfolds too much, it could potentially kill the cell. Now, I can't think of any examples of an organism that you can have burning in your house that will not cause a fire, because my mother never let us play with fires or with matches. What else? I think we're done here. So let's move on. There are different types of halophiles. There's an obligate halophile, and this cell requires high salt concentrations to grow. An example would be a bacteria growing in the Dead Sea between Palestine and Jordan. And there are facultative halophiles. These do not require a high salt concentration to grow. In fact, they tolerate and grow in 2% sodium chloride or less, and some can even grow up to 15% salt. And a hypotonic solution, one that has low osmotic pressure. There's more water outside the cell than inside the cell, so water will enter the cell by osmosis. And if too much water enters the cell membrane will expand until it pops and then leaks out its cytoplasm, and that will kill the cell. Any questions about any of this? All right.
What was hypotonic again?
Where there's more water outside the cell than inside the cell. And so water will move into the cell. Now, the tonicity is not defined by water. It's actually defined by the particulate or the particle matter, meaning the elements dissolved in the cytoplasm or in the liquid that the cell is in.
Okay, thank you.
A bacteria has a cell wall, so it may not lyse because the cell membrane will just come up to the cell wall and then stop expanding. And that's how cell walls protect their cell. All right, carbon's the next chemical requirement we need to talk about. All living things require a carbon source. Half of the students in the microbiology lab will work on this, or will answer the question on this, or you will be maybe a little bit unlucky if you don't ever work on it in the microbiology class. You will probably run into it when you're working in a clinic. Carbon is typical of all cells. Half of cells is carbon with the water removed, meaning we're measuring carbon under the dry weight or the dry slide. And how you remove the water is you briefly put water on the slide, or you put the slide in the water, and then you rinse the slide, and that will prevent cells from getting carbon if they require carbon. Photosynthetic organism, it does require carbon, but it can get it from CO2 in the air, so your treatment will not stop the carbon from moving in or the water really from moving out. And that's just because the windows are open or they're together and they're just staying that way. So carbon, about half of it is the typical weight of a cell. Carbon units are used in structural and organic molecules, and carbon is, of course, used as an energy source. When we want a quick and healthy diet, and you want to eat the carbs, you can eat it, the fancy carb, I don't know the name of them, sorry, I don't buy it, but you can buy the fancy carb from one of the stores or something like that. We have heterotrophs. They use the same organic carbon source as the heterotroph uses for getting its energy needs from. So with heterotrophs, the carbon source is equal to the, what is that called? I'm losing that word again. So heterotrophs use our organic carbon source, and autotrophs use carbon dioxide for their carbon source. Any question about any of that? All right, some other chemical requirements are nitrogen, sulfur, and phosphorus. All living things require nitrogen, and nitrogen is about 14% of the dry weight of a cell. So if you wanted to determine the amount of nitrogen in the cell, you could actually weigh it up with the dry weight and take 14% of that weight, and that's how much of that weight is in nitrogen. Nitrogen is found in, of course, in amino acids and proteins. It's also found in DNA, RNA, and ATP. Nitrogen is often obtained from the decomposition of proteins. So if you want nitrogen, you could just start degrading one of your proteins and you will get a nitrogen source. It will be attached to an amino acid, but another reaction can remove the sulfur compound from the nitrogen compound. Many bacteria do get their source of nitrogen from protein. However, many bacteria also get their source of nitrogen from either ammonia, NH4 plus, I guess that's ammonium, and nitrate, NO3 with a negative charge. So these are bacteria that can use nitrogen containing compounds as a nitrogen source. A few bacteria can even use N2 gas, nitrogen gas, that is in the atmosphere. And when they do, they fix that gas that was once in the view, and now the nitrogen can be seen in the bacteria or in some product that the bacteria makes. And that happens with more than just nitrogen. Sulfur can be found in amino acids, the vitamins, B vitamins, thiamine and biotin. Sulfur can be found in DNA and RNA. Sulfur is obtained from decomposition of proteins in most organisms. There are a few that can obtain the sulfur from eating mushrooms, I guess raw. Sulfur is another element that our body must have in certain amounts. One of the important things that sulfur does for us or for a sulfur bacteria is sulfur gives the cell the ability to transform the cell into another washcloth that is illustrating the sulfur requirements, and you're looking at different sulfur requirements. Many bacteria can use sulfate, SO4 with a -2 charge, or hydrogen sulfide gas, and some can even use sulfur directly from a sulfur spring or sulfur in the soil. Any questions about any of that? All right, phosphorus is another chemical that is required by all living things. You can get phosphorus from DNA, from RNA, and from a portal on a chart. Yes, DNA, RNA, ATP, membranes, and some proteins have phosphorus in them. Any question about any of that? All right. Trace elements are also required by all living things. Trace elements are inorganic elements required by microbes in low amounts. Often, trace elements are required as an enzyme cofactor. So the enzyme can work, but only when it is being helped by the coenzyme, the cofactor. Some examples of trace elements include iron, copper, molybdenum, and zinc. We require all of those in our diet. Fortunately, for most of us, we already have enough in our diet, and so we'll be healthy as long as we're eating. Any question about any of that? Organic growth factors are another chemical requirement of all living things. If a species of bacteria requires an oligogametophyte, meaning a child, for an experiment, you'll be rough how to do this in the lab, and it isn't explained in the lecture because the lecture is referring to a previous scientist. And what we're doing here is trying to tell you when organic growth factors were first obtained, it quickly was learned that some cells absolutely require these organic growth factors. And if you don't add them in one way or another, the cells will die. Any question about any of that? Some of the examples of organic growth factors that are required by some living things include various vitamins. Anyone know a vitamin for us that is an organic growth factor? Come on, there's only about 10 vitamins.
Vitamin C?
Yeah, vitamin C is a very good example. We require that for an organic growth factor, and we require it also as a protein to help degrade usually toxic proteins. And I'll talk more about that in a little bit. I mean, we're not going to discuss that because we're limited on what we can cover. When we're talking about oxygen, it can also be a chemical requirement that all living cells require in one way or another. The living cell may want no oxygen, or it could want low oxygen, or it could want, I don't know, medium or regular oxygen, and it may require oxygen at higher levels than what is in the air. Those are just some of the possibilities for oxygen. When we're talking about oxygen, it's important to realize that oxygen is, at least in one way, a toxic substance, even for us, and we require oxygen. However, when we make oxygen, you tend to put it in, and then the socks will just slide all over. So if you need your socks to be in one place, you shouldn't put in this pad that prevents the bacteria from sticking, but you get to be stuck onto the pad. So oxygen is toxic even to us if we were to get too much, and if the wrong reaction were to happen, we would get too much oxygen, and that would make us sick and eventually die. Let me briefly talk to you about a story, and that is, it's probably been a couple of years now. I was reading, and the doctor didn't think it would be a problem because the patient had most of the parts or enzymes to make oxygen, but there was one either defective gene or one gene was not working, and it was the good gene. So the patient only had one gene, which was the defective gene for handling oxygen. And because of that, some experiments that were run had to be discontinued because the patient was so sensitive to having lower oxygen levels than normal. It wasn't low oxygen because that would have killed the patient, but it was fairly low. Any questions about any of that? All right, there's 4 toxic forms of oxygen. There's singlet oxygen, and that is when we have an oxygen molecule and the electron becomes boosted to a high energy state. This makes singlet oxygen extremely reactive, and it can occur in the phagocytic white blood cells, but usually the phagocytic white blood cells will use singlet oxygen to help defend the body against a foreign invader. Any question about any of that? The second toxic form of oxygen is superoxide free radicals. Free radicals are always bad, but we can have a superoxide free radical. When a superoxide free radical becomes a free radical, meaning it's lost an electron, it'll want to regain that electron, and it will usually steal it from whatever that molecule bumps into. So a superoxide free radical tends to steal electrons from other molecules, and that could, well, it does turn that other molecule into a free radical, which can then steal electrons from other molecules, and it eventually will damage the DNA. Superoxide dismutase, or superoxide free radicals, are removed by the enzyme superoxide dismutase, or SOD. And so we have a superoxide free radical, actually 2 of them, 2 hydrogen ions with SOD produce hydrogen peroxide and oxygen. So in our cells, when we make superoxide free radicals, which we do every once in a while in aerobic respiration, we do, and then we have the enzyme SOD to take care of something we should have never made. Any question about any of that? All right. The third toxic form of oxygen is a peroxide anion, shown here, and it's an oxygen gas molecule with 2 extra electrons. Hydrogen peroxide is an example, and it is neutralized by 1 of 2 enzymes. Most cells contain catalase, and catalase will react to 2 molecules of hydrogen peroxide to make water and oxygen. And then we can see this by observing the bubbling forming in the hydrogen peroxide. Is there a question? Okay, somebody has your microphone on again, so please turn your microphone off if you aren't going to be using it. The other enzyme which can convert the peroxide ion into something non-toxic is peroxidase. It can combine hydrogen peroxide and 2 hydrogen ions into 2 water molecules. Most cells have catalase, a few have peroxidase, and a few have both catalase and peroxidase. We're talking about different microorganisms. Our cells have catalase. Okay, the 4th toxic form of oxygen is a hydroxyl radical, and it is a OH group with an extra electron, and that's different from the hydroxide ion, which is OH with a negative charge. This one has no charge. And of course, it's different than the OH group on alcohols, but in a sense, it's similar because it has the same atoms. It just has a different charge. Like OH minus is hydroxide ion. It's not toxic to the cell. Hydroxyl radicals are toxic to the cell, and the reason is whatever molecule this molecule, the free radical, bumps into, it'll steal an electron from that molecule. And that could potentially lead to a chain reaction of one molecule stealing an electron from another molecule, stealing it from another molecule, etc. Hydroxyl radicals are performed by aerobic respiration in trace amounts. It's also performed by ionizing radiation. And then hydroxyl radical is made whenever hydrogen peroxide reacts with a certain metal. So you don't want to get a metal around if you're making hydrogen peroxide, for example. Antioxidants remove this hydroxyl radical and other free radicals. And then I just want to mention two antioxidants include vitamin C and vitamin E. They are antioxidants and they can remove these molecules from the cell. All right. So we kind of hinted at this, that oxygen can be a requirement for cells. It certainly is if the cell is an aerobe. It must then survive with oxygen. And when oxygen will become absent, the cell that's an aerobe will die. Aerobes, as you recall, produce more ATP from a molecule of glucose than any other mechanism for making ATP. So aerobic respiration is the most efficient, and microbes are classified on the basis of the effect oxygen has on their growth. Let me blow this up. If the microbes will only grow at the top, so it has the most growth at the top, and as you go down the tube, there will be less and less growth. This is a aerobe, in fact, a strict aerobe, because it's only growing with oxygen. And most growth will be at the top of the tube. The growth will decrease. And then at a certain level, low down in the tube, you'll get no oxygen. And the reason being is there's no oxygen around to burn up that molecule. A facultative anaerobe is one that grows best at the top, but as you go down the tube, you get less and less bacteria. There is a slight difference between a normal tube of media and then a tube of media where we're checking to see if it's a facultative anaerobe. Here we're talking about facultative anaerobes, and they grow best at the top of the tube. And as you go down the tube, you'll get less and less growth, but you will get growth all the way down the tube. And that's just because what a facultative anaerobe is. It can grow in the absence of oxygen, but it can grow in the presence of oxygen. And if oxygen is available, the organism would like to grow with the pizza method or the oven method because it's less energy consuming than something like a gas grill or, I don't know, convection or oven. If, on the other hand, the growth in the tube only grows at the bottom of the tube, then it is an obligate anaerobe. And the further up you go in the tube, you'll get less and less growth. So there should be a 1 dot there. And then above that last growth that you can see, you'll see no further growth. And that's because there isn't any. There's oxygen in this region. And so the obligate anaerobe cannot grow. The aerotolerant anaerobe is one that grows evenly everywhere in the cell. The reason why it grows evenly is it is an anaerobe, but it can tolerate oxygen. It has all the enzymes it needs to detoxify the toxic substances of oxygen. And there's like 4 of them, 4 toxic substances, 1 for each of our labs. The last one is a microaerophile, and it is an aerobe. So you must supply oxygen for it to grow, but it does not like to grow when oxygen is at its highest level, which would be 20 to 21% at the top of the tube. It likes to grow at reduced oxygen levels, and that would be someplace in the middle of the tube, because at the bottom would be an anaerobe, and these aren't that. They are an aerobe. It's just that they are an aerobe that does not want complete oxygen. It will not grow well at 12% oxygen. All right, so that tells you the effect oxygen can have on the tubes or on the growth of the bacteria, and it just depends on what the bacteria species is. Let me talk a little bit more about the tubes. This is a tube containing thioglycolate, and when you make your thioglycolate, it will remove the oxygen from the media. But once you take the tubes out of the autoclave, oxygen will come from the air and slowly work its way down the tube. At the top of the tube, by the time we use it, the top of the tube will have full oxygen, and that would be 20 to 21% oxygen. As you go down the tube, you get less and less oxygen until you reach the bottom of the tube where there will be no oxygen. And we can use a thioglycolate media to test where the bacteria grow and what the bacteria is: an obligate aerobe, a facultative anaerobe, or an obligate anaerobe. Any question about any of this? Unfortunately, we won't be in the lab because we used to do tests like this where you would do it yourself. Maybe some of you will do this test on your unknown because it does provide useful information. All right, are there any questions? If not, it's 6:20, so I think I'll stop here and I will continue at 6:30 with the lab. Bye.